Alright guys, this is going to be your Chapter 12 review video, and this chapter covers inheritance patterns and human genetics. So this one, now earlier in the year we talked more about Mendel and his side of genetics. Now this one focuses a little bit more on the human side of things. So first we want to talk about our chromosomes. Now our chromosomes, there's two different types that we have. We have sex chromosomes, which are chromosome number 23, and they determine the sex of the individual. Now, we're going to indicate these with a purple color here. Now, our sex chromosomes are chromosome number 23, and they're located, they are the last ones. Now, in this case, these chromosomes are an X and a Y, therefore this will be a male chromosome, but the sex chromosome's always the last set. Now, our autosomes are every other chromosome in our body that are not the sex chromosomes. So in this case, these are chromosome number 1 through 22. So we're going to indicate these by green. So our autosomes are everything you see circled in these green letters right here. So the autosomes, they are controlling the majority of the traits in your body. And the only thing that the sex chromosomes indicate are what the sex of the individual is, male or female in that sense. Now our sex chromosomes, they're a little bit different. So males have sex chromosomes X, Y. Now you see these chromosomes down here, and you'll notice that the Y chromosome is a very small, very short chromosome. And the reason for that is really the only thing that carries on this is this little gene that usually is located right there. And that is the SRY gene, or the sex determining region Y. So this region right here, located on the Y chromosome, and this gene, it codes for a protein. And what this protein does is it causes the formation of the testes. In other words, this little gene right here is what makes males develop into males. Now, you always hear the saying that males and females develop the same initially. Once this SRY gene kicks into place, that's what codes for all the things, all the proteins that make you into a male. Now, the females, their sex chromosomes are XX, and you can see those right here. Again, two long chromosomes, therefore females XX, males XY. Now, next we want to talk about our linked genes. Now, linked genes are just saying that where their genes are placed. Now, the only special ones that we usually talk about, because usually when they're on 1 through 22, we just say autosomal, are X-linked, which are located on the X chromosome, or Y-linked, which are located on the Y. So these two chromosomes, now the Y really doesn't have anything on it. The only thing that's on there is that SRY gene for males. Now X chromosomes are things that are carried on there are things for like color blindness. And another example that you may see is hemophilia. Now these being carried on the X chromosome, the female will have 2 copies of those. Okay, because they have 2X. Now, if they have one dominant, one recessive trait for that gene, they're obviously okay because it's a recessive trait. Whereas males, if they get the recessive trait on their 1X chromosome, that's what is going to be expressed. So this is why sex-linked or X-linked genes are found in males much more than females. And pretty much sex-linked, all we're talking about here is anything that is located on our sex chromosomes. Okay, so again, X-linked, Y-linked, the only thing Y-linked, SRY gene, making males males. Now, we refer to linked genes. We're pretty much just saying that these are two genes that are very closely related or closely associated with each other. Okay, so these are genes on the same chromosome. No, I'm just going to put chromo. On the same chromosome and are very close. In other words, when crossing over occurs, and crossing over is just when the DNA is mixed by pieces breaking off and reattaching. Okay, so what we're saying is they're very close, and when they cross over, those genes usually don't split from each other. So two genes that are linked, like gene 1 and gene 2 down here, you notice that these genes are very closely related. So if it's very rare that the chromosome is going to break between them. Now, genes on opposite chromosomes or homologous chromosomes are not linked, or genes that are very far apart are as well as not linked. They must be very close together. So how we measure this is by looking at the crossing over or the homologous data in the F2 generation. Now, in the F2 generation, we look at this data, we can tell how close they are related or how close they are together. So this is where chromosome mapping comes into play. Chromosome mapping just shows the distance between two genes. Now, how we measure these are these things called the map unit. And the map unit is when we have that F2 generation, we look the recombinant data of these two genes. So in other words, how often are they associated together? 1% of them being recombinant or linked together in the generation equals 1 map unit. So for example, we see down here, body color and wing size. They are 17% of the time they are associated with each other. What that says is that is 17 units apart from one each other on that chromosome. Okay? So the percentage that they are associated with, the closer they are together. Now, mutations. Mutations occur in our body. Mutations are just a change in the DNA of the organism. Okay? Now, in this sense, mutations can cause a few things. They can be helpful. In other words, they help the organism survive better. They can be harmful. Now, these are things like cancer or maybe some sort of genetic disorder. Or they could be neutral, where they really have no effect whatsoever on the organism. Helpful mutations drive what's known as evolution. Harmful is what leads to death and cancer in individuals. Now, chromosome mutations, the thing about these is this is where our chromosome is rearranged, or a piece is gone, or a piece is added. So we have a few different types of chromosome mutations we have. The first one is deletion, where a loss of a piece of the chromosome occurs. A loss of a piece of chromosome. So in other words, we have this piece of chromosome right here. That piece of chromosome is eliminated and the chromosome now becomes shorter. Now the thing about these is chromosome mutations are much more dangerous because they code for multiple genes at one time. Now inversion, a segment breaks off. And when that segment breaks off, it flips and reattaches. So in other words, imagine like you driving backwards down the road. It doesn't make sense. It becomes very dangerous. So inversion is right here. So you'll notice this piece right here, big, long yellow, light yellow, short yellow. So you see how those two flip locations, and it can cause a misreading of our genes. Now, translocation is where a piece breaks off and attaches to another chromosome. So in translocation, you see the piece of the red, the piece of the yellow break off, and those two pieces switch with each other in translocation. And finally, duplication, where we have a repeat segment of the chromosome. Okay, and in duplication, you kind of see this right here. We have this piece is duplicated twice. Now we have a double copy of those. And again, chromosome mutation cause a lot more problems than just gene mutation, because gene mutation, or chromosomes, have multiple genes in them. So nondisjunction is pretty much when a chromosome fails to separate properly. So it's the so those chromosomes, they fail to separate. I can't type here. They fail to separate during meiosis. So what this leads to is something called Down syndrome. And this is where we have, instead of our normal 46, we have 47 chromosomes, also known as trisomy 21. So where we have 3 chromosomes at the 21 spot in our karyotype. So what happens here, during meiosis 1, we separate properly. Now during nondisjunction, these 2 blue chromosomes don't separate and go 1 to each one. Both of them come to one of the gametes, and what this does is leads to an extra copy of our DNA. Now, Down syndrome is a genetic disorder caused by 3 chromosomes at the 21 spot. Causes a lot of mental problems for the individual that contracts this trait. Now, gene mutations are when the nucleotides are changed within our DNA. So the different type of mutations we have substitution. And this is where one nucleotide replaces another. So in substitution, as you see from the picture over here, we're looking at this segment right here. So our normal strand is GAA. Now in our new strand, in our substitution, we replace that G with an A, and we have AAA. So what we get is, instead of a glucamine, we get a lysine. So our amino acid chain is changed. In deletion, one or more nucleotides are deleted or eliminated, are lost. Okay, so one or more nucleotides are lost completely. So in this case right here, our deletion, we had our A right here, and what happens is we take that A out. Now what this does is it causes a frame shift, and what that means is, when we normally read, we read in these 3 codon sequences. Now when we frame shift that, all of the letters get shifted down 1, so it changes our amino acid change, and we get 3 amino acids that are not correct. Insertion is when one or more nucleotides are added. This kind of does the same thing as deletion. So in insertion, we add a piece there. Again, we're frame shifting our reading down. And frame shifting is just moving our 3 codon sequence in that scenario. Okay? Now, the next thing we want to talk about is a pedigree. Now, a pedigree, what it does is it looks at a characteristic or a trait of a whole family. So it's like a family tree, except it's tracking one genetic trait, okay? So when we look at a pedigree, we have females, which are indicated by circles. We have males, which are indicated by squares. Now, when they form a mating pair, we throw a line in between it. When they have kids, the line comes straight down and it branches off of the two, okay? So for example here, these two individuals in generation 1, which is indicated by Roman numerals, okay, gave birth to 2 children in generation 2. Now, if they get married, just a line comes off of there to indicate that that marriage has occurred. Now, in these pedigrees, if you see something that is completely shaded in, that indicates an infected individual, okay? So, for example, this right here would be an infected man, okay? Now, you can also have ones where they're half shaded in. Now, the half shaded in ones are what are known as carriers. And a carrier is just when we carry the information. So if I have, let's just say, genotype capital A, lowercase a. If this is a recessive allele, this individual will not express that recessive trait, but they carry the opportunity to pass it on to the next generation. So we have carriers, we have infected individuals, and we have healthy individuals. Now, when we are showing these inheritance patterns, we look at autosomal compared to sex-linked. So an autosomal trait will be expressed equally between males and females. So in other words, males and females, they get the trait in an equal number. So if you look over here, autosomal dominant, both, equal number of males and females getting it. Autosomal recessive, just one. Doesn't have to be a man, doesn't have to be a woman, just what we're looking. Now in sex-linked traits, this occurs more in male individuals. And the reason for that is because males are XY, and usually sex-linked traits are carried on the X chromosome. So because they only have one X, if they have the recessive trait, they are going to express it. So we look over here in our X-linked recessive, you notice only males are the ones that are expressing it. Now the females are the ones that will be carriers for that trait. So let's look at a couple different patterns of inheritance that humans do express. And there's a few of these, so I'm just going to kind of go over them very quickly. Now polygenic means that there's multiple alleles per gene. So what that means is, for example, in the color of your skin, a light individual, skin color is 6 alleles make it up. The more that are recessive, the lighter your skin color will be. The more that are dominant, the darker it will be. And then there are multiple different patterns between it. So polygenic traits are things that have not just 2 genotypes, but they have multiple different genotypes within that. Now, a complex individual or a complex type of inheritance has to do with genetics, your genetic traits, as well as environmental factors. So in other words, cancer is a very good example of this. So somebody who has cancer run in their family, you're said to have a stronger chance of having cancer because it runs in your family, because of your genetics. But it also has to do with your environmental conditions. So do you smoke? Do you drink? Do you eat processed foods? That increases your likelihood of having cancer. Now, multiple alleles are when genes with 3 or more alleles, okay, they have 3 or more alleles within them, and they control the trait. So this would be like ABO blood typing. So for example, we could have dominant A, we could have dominant B, or we can have the recessive trait, which is what codes for the O blood. Okay, and those different characteristics are listed down here. O blood, as you kind of got cut off here, 2 recessive alleles. A blood, you can have 2 different. B blood, 2 different, and then AB blood from those, as you can see. Now, codominance Codominance is when both alleles of an individual are expressed. So, for example, I just used a 4 o'clock flower. We have a red, we have a white, and when both of are expressed or heterozygous, it creates a pink. Now, blood typing is like this as well, especially when we get the blood type AB. So, when you have a copy of an A, copy of a B, both of those are expressed. Both those proteins are on your blood, and you have AB blood. Now, incomplete dominance is when you have, a mix between the two, or an intermediate between the two parents. So an example of this would be when you have straight-haired mother plus a curly-haired father. What you get is a wavy hair individual, or a combination of both the traits. And the last one we have on this page are X-linked. Now this is a disorder carried on the X chromosome. So in this case, males are going to contract this much more than females ever would. Okay? Now the next two, we have sex influence. Now sex influence traits are like males and females have different phenotypes for the same genotype. Okay? Now in this case, it's male pattern baldness. So what the characteristics should be up here, we're going to have, when you are normal hair, you have our homozygous dominant. So we're going to call this capital B. Let's go the other way. Yeah. So bald would be the intermediate. So in other words, baldness, I put that the wrong way. Baldness is dominant in males and recessive in females. So in order to be bald in a female, you must be homozygous recessive. So in other words, heterozygous individual, capital B, lowercase b. A male will be bald. A female will have normal hair. So there are two different characteristics for what we have. And a single allele trait just means that one dominant trait controls expression. And the one that we talked about here were Huntington's disease. And Huntington is one of the genetic disorders that you will have to know for the upcoming test. Now, finally, what we have are genetic screening and genetic counseling. Now, what a genetic counselor will do is they will perform a genetic screening on you, or they examine the person's genetic makeup. So they look at your genes and they give you opinions on what you should do in the future. So do you have a genetic disorder? Should you have kids? Things along those lines. Now, what they can do for a genetic screening is they could do things like they can look at your karyotype, or they can perform a blood test to look for specific proteins. They can also test your DNA directly, and this is just determine whether you have these traits or not. Now, genetic counselors, their job is to inform you on their genetic makeup, or on, obviously, And what they really do is they have a tough job. They have to determine, is it their responsibility, it's their responsibility to say, listen, you have a good percent chance of passing on that genetic trait. You probably shouldn't have kids, or your kid will probably have that genetic order. It's a very tough, very sensitive job that they have to do. So this is your review for your Chapter 12, Inheritance Patterns in Human Genetics. Hopefully this helps you out for your test. This is Mr. O'Brien signing off.